We have two major swabbing sites, one each at the Winchester and Basingstoke hospitals, in addition to multiple other smaller testing sites across Hampshire. The swab, labels and packaging are prepared, staff put on their personal protective equipment, and the patients arrive for testing at these sites and are swabbed by our dedicated staff. Once the swab has been taken and the waste has been disposed of safely, the swab is transported to one of our 4 COVID testing labs. There are multiple ways that this can happen, such as via porters and couriers. To date, over 65,000 samples have been analysed at our labs since spring 2020, when the pandemic began. When the sample arrives at microbiology, they are handed over to our reception staff, who ensure they reach the COVID testing area of the labs quickly for processing. The samples are then placed into large containers for storage in preparation for testing. When we are ready to process a sample, it is given a unique barcode for identification and then entered into our pathology-specific IT system using the ICE number on the ICE sticker. This allows us to keep track of which samples we have received and when, and what stage of the testing the sample has reached. At this stage, a small container called an Eppendorf tube is also labelled with this unique barcode, and this container is used for much of the testing. The barcode is also put onto a run sheet, which details which samples are being processed at which time. All of this labelling helps to ensure no mix-ups occur between different samples. This stage of entering the data into the IT system and labelling the samples is a full-time job for 2 people 7 days a week. Each sample and Eppendorf tube are placed into separate racks of 46 samples, which is the number of samples tested in one run in the lab. Once the samples are prepared and labelled, they are inactivated. This is done in a safety cabinet or hood, which protects the staff member from the virus in the sample. Inactivation destroys the virus so it cannot infect anyone, but ensures the genetic material is not damaged, so parts of the virus can still be detected using our tests. A small amount of the fluid in the bottom of the sample tube is removed and pipetted into the Eppendorf tube. This tube is then heated to ensure it is safe for further testing outside of the safety cabinet. And the original sample is saved in case there are any problems and the test needs to be repeated. Each sample then needs to have the genetic material of the virus extracted from the sample to help ensure an accurate result, and we call this the extraction stage. Each sample is loaded into a machine called the Maxwell, which performs this process automatically. Each sample has a strip of reagents and a tube of purified water added, which the extracted genetic material is put into by the end of this process. Once this preparation is performed, the samples are loaded onto the Maxwell, and this stage takes about 30 minutes. Once this is complete, the purified genetic material is ready for the final test. The purified genetic material is then pipetted into a series of tubes called a lamp strip. Each sample is tested twice to help ensure an accurate result at the end. At this stage, a solution called the master mix is also added. It contains all the components needed for the genetic material to be copied multiple times and detected using our final analyzer. The lamp strip is then flicked to mix the reagents and spun to ensure all the fluid is at the bottom of the tubes ready for the final step. The strip is then loaded into our final analyzer called the Optigene. This detects the virus by copying it multiple times and uses fluorescence or light at specific wavelengths to determine if it's present or not, and this takes about 20 minutes. This graph shows the results in real time, and we can see that one of them is increasing significantly and quickly, and this represents a positive result. Each result is then written onto the run sheet we prepared at the beginning of the process. This includes information such as when the sample became positive and if the sample was positive in both tubes. If it is only positive in one tube, we repeat the test to ensure the result is accurate. Positive results are assessed by the microbiology consultants, and the site team are informed so the result can be actioned. Each result is entered manually into the pathology IT system by one of our scientists. This result is then sent back into the IT system automatically so it can be viewed by the clinical teams. This whole process takes about 3 hours from the inactivation stage to the final result. This information is then imported into a different IT system, which converts this result into a text message. We hope this has helped you to understand the complex nature of COVID testing at Hampshire Hospitals, and thank you for your patience during this challenging time.